Before we begin the presentation, I would like to introduce today's speaker, Dr. Bernd Wendt. A full speaker bio can be found in the blue widget at the bottom right of your console. Bernd has more than 15 years of experience in the life science industry, specializing in molecular design and data mining techniques. Before joining Certara, he headed the molecular and PK modeling lab for Elara Pharmaceuticals, a spinoff from the European Molecular Biology Labs. At Elara, he focused on the development of new oncology therapies. Dr. Wendt earned his PhD in chemistry and biochemistry at the Philipps University in Marburg, Germany. Bernd, welcome to today's event. I'll now turn it over to him to begin today's presentation.
Thanks, Suzanne, and welcome all attendees to today's webinar. It's always a pleasure to share some tips and tricks around Phoenix to a live audience. In this webinar, we will focus on Phoenix workflows, where we share an application in the area of preclinical development. As you can see from the title, we will deal with estimation of preclinical PKD parameters for anti-cancer agents. I will be showcasing a workflow that I've developed on data from Elara Pharmaceuticals, a company I worked before joining Certara. The goal of this webinar is to give you some ideas and perhaps also some motivation to apply these tips and tricks to your own data. And I hope this will be of interest to you. So we are starting with a fact sheet on the front runner compounds of our multiple myeloma program. This is a condensed list of all important properties of our compounds. You see there are in vitro data, in vivo data, and PK/PD data. This fact sheet is used to give you a quick overview and should help you to quickly select the top-ranked compound that you may want to move into preclinical development. And you see there are 3 compounds. These are the front-runner compounds. They are compared with their figures against a competitor compound. But this slide is really too busy, too many figures. Let's focus on those parameters that are relevant for today's discussion. So what you are seeing here now in bold black are the PK parameters like half-life, Cmax, and AUC, as well as PK/PD data. All these parameters can be estimated by the workflow that I will show in this software demo. And that is really important because, as you can imagine, there's lots of data behind these figures. And also, new data is being generated on a continuous basis. So it's very important that your modeling activities will keep pace with the development of the compounds. But there's also another point here. When you look at the PK/PD data, you see the results from the xenograft studies where the potency of the compounds are expressed as tumor growth inhibition. This is really not the best way to measure the potency of the compounds when you compare across compounds. And I will explain that on the next slide. So when doing preclinical experiments in rodents, the most widely used efficacy endpoint that is used is a percentage test to control of tumor weight ratio. This is typically also the figure that you will get from your CRO as part of your study report. But sometimes also the median time to reach a defined tumor weight is used. It should be kept in mind that those figures are dependent on the experimental conditions. So those values will change when you change the dose regimen, and the time. So a better way to measure the potency, specifically if you want to compare across compounds, is our recommended solution would be to use models, PK/PD models on xenograft study data. Because this provides you with a quantitative link between the plasma concentration of your compound and the respective tumor growth inhibition. And this is really independent of the experimental condition. And also, if you use a mechanistic, or like we do in our software demo, a semi-mechanistic model instead of an empirical one, you can derive parameters that help you explain the biology. And another benefit of using PK/PD models is later on the ability to predict effects in humans. Let me re-emphasize once again the effort it takes to work through the different studies. And here we are dealing with lots of studies. So first there are the chemists that come up with new compounds on a daily basis, and many of those need to be studied, and they have to be studied. And also there are the biologists, and they keep changing the study protocols by maybe changing the dosing regimens and so forth. Also, they don't want to make your life easier when they are not focusing on a single cell line, but typically they use different xenograft models. And later on in the process, when you are close to the clinical stage, you have to deal with different formulations for the same compound. So this gives rise to lots of different studies. And as a small biotech for Lara, we have outsourced all in vivo studies, and so they were run by CROs and not a single CRO. Typically, you deal with a few of those, and every other CRO has a different output format where they put their reports in. So this makes life really, really hard. Finally, you also have to deal with different types of analysis. You deal with in vitro data as well as in vivo PK data, and finally also the xenograph data. Overall, I think you can see the challenge that it is actually very difficult to keep pace with all the updates and the progress of the program. I mean, and with your modeling activities, you really want to support mission-critical decisions like updating the fact sheet that I showed in the beginning. Here again, I mean, our recommendation is around Phoenix WinNonlin workflow. So this is really the way to move forward to save the time. So this, what you can see here is a display of the workflow in the Phoenix interface. Phoenix workflows allow you to organize your data and to help you with the process of generating a full PK/PD profile. You set up the workflow step by step, and then you can save it as a template and can reuse it at a later time whenever you receive any updates or when you have to deal with a different study or a different compound. And this will actually save you a lot of time. In my software demo, I will present a compound-specific workflow. It is the one that you can see here in the picture. We start by importing the study data and then run through some preparation steps, so make the data ready for the modeling stage. And before that, we will generate some exploratory plots in the exploration part, and then we look into how to model the data. When finished with the modeling, we will also simulate different dosing regimens to help refine the study protocols and so forth. And at the end, I will show you how you can efficiently report about all the different steps in the workflow. So in the first workflow, we will look at a single compound. And briefly in the second workflow, I will show you how you can compare several compounds in one workflow. So let me switch over to the software demo. So to go to Phoenix. So this is the Phoenix interface. You see it's exactly the same workflow shown here in the main window of the Phoenix interface. And on the left-hand side, you see the structure of the project file. It's for a single compound, EL103. And the first step we are going to, and this is a typical first step in the project, is to import study data. And here we have three different types of imports. First of all, the PK studies, where we have two different routes, the IV route, and here's the sheet for that. You've got the time, and for each of the animals, the concentration in separate columns. And the same with the PO route, same format, but this has to be rearranged to be used for Phoenix analysis. But we'll come to that later. So these are the PK data. Then what I typically do is I import some prepared timetables to help with simulation studies. So for example, I just put in the time points for a 28 QD study. So these are just the time points to prepare dosing tables to be used for simulation and modeling. The same you can also use, for example, you can easily switch between a QD and BID dosing regimens. So I've got a template here for BID. And then there are also other different dosing regimens like a QD study, 7 days on, 7 days off, and other variants. So these are just very helpful in automating several steps in creating dosing tables and running simulations. So this is about simulation, and then we come to our PK/PD data. This was a tumor growth inhibition study in a breast cancer cell line, and these are the sheets that we've imported from that. But let me first show you how the data looks like in Excel, how we received it from our CRO. You see this is an Excel workbook with lots of worksheets. And the summary sheet that is shown here gives you an overview what has been done in this study. So we've got 8 groups. The first one being the vehicle, the control group, where we just measured unperturbed tumor growth. Then we've got several compounds. Among those, one of our frontrunner compounds, EL103, where we measured tumor growth at 3 different dose levels. So this is some information that is very important for Phoenix analysis. So this summary worksheet needs to be imported. But then we also have this tumor volume sheet. And here you find detailed information about the tumor volumes. There were 10 animals in each group, and tumor volumes measured on those days over the whole 48-day study period. So this is Excel, and how does it look like when it's imported into Phoenix? Let's go to the summary sheet first. So this is the imported summary sheet, and it looks much the same as in Excel, same column headers and so forth, but it has to be prepared a little bit in order to be used for Phoenix analysis. And next are the tumor volume data, yes, that you can see here, also much the same as in Excel. So these are very simple steps. You just import from Excel into Phoenix and get a copy that you can use here for your workflow. So this was your study data. Let's go to the workflow, where we got the different sub-workflows. So we start with preparation. In preparation, we have 2 different types of imported study data, PK data and PK/PD data, which are handled separately. So the first step is to append the IV and PO records. So you've got one single sheet over all concentration time profiles. But then we have to stack up the concentrations, because Phoenix requires you to have just a single concentration. So you've got all the animal data stacked up, and you end up with one concentration column. Then you need to adjust some settings here, like, for example, you want to change the units from nanograms per mil to micromoles per liter, as well as the dose accordingly. And then for later, for modeling purposes, we actually have to split up into the routes once again into IV and PO data. And then for our exploratory plots, you just run a descriptive stats on the profiles, so you get a mean profile for the IV and for the PO route. And that's the preparation of the PK data. You don't need anything more. I mean, this is everything you need to do in order to get the data ready for the analysis. There's a few more steps in preparation of the PK/PD data. We have 2 sheets. First, the summary sheet. Here it's important to have the right group assignment, because that determines the dose level and dose amounts in this study. This has been done in this step. Then for all the 4 sheets of the tumor volume data, you just add them up on top of each other. So you append 4 sheets, and this is a resulting tumor volume sheet. And as with the PK data, you just need to stack them up so that you have a single tumor volume column for your analysis. And next step is also doing some adjustments here in preparation for the next step, where we actually want to, and this is really the key step, where you join the summary sheet with the tumor volume data. And this is important because you have to, for each of those records of the tumor volume, you assign the right dose level and dose amounts. This is very important. This next step is maybe not required, but it's very useful for modeling. So I'm just filtering out the control data, the vehicle data, where no compound was applied, to be separately modeled. And for the next 3 steps, we are actually preparing the dosing table for modeling. So first of all, you select one of the prepared simulation tables. See, here's the QDay 28-day study, and you join that with the tumor volume data to get this kind of dosing table, the first step. Then you need to join the dose amounts and the compounds, and finally do some adjustments and finalize the dosing table. And this can be directly used in the modeling stage. And as with PK data as well, we generate some mean profile data also for the tumor volume data. So we got some mean profiles here. And next step is also more optional. It's creating just extracting the time points for tumor volume measurements, and this is also required for some modeling when we try to simulate some different dosing regimens. So this is all what we need to do for preparation. Everything is now ready for the modeling stage. But before moving into modeling, we just create some exploratory plots to make sure that all the previous steps, the import and the preparation of the data were done properly. So you look at those plots and see, okay, this is the mean profile for IV and PO route, plus the observations in red circles. Looks good. But also you can create some individual plots of your profile, like this one here. This is for the PK data. And now switch over to the PK/PD data. Here you've got all the 4 curves for the compounds at the 3 dose levels, plus in green the control, the vehicle. And again, here some more details where you got the mean profile together with the detailed tumor volume measurements for the 10 animals per dose level for all these 4 groups. Okay, so this looks good. So we are now ready to start into the modeling stage. And here actually we have to deal with 3 different things. As you've seen in the fact sheet, there were some PK parameters required, and this is typically what we do, what we can get out of the non-compartmental analysis. So we run an NCA analysis, and this is what you get out of it: half-life, Cmax, AUC last, some volume and clearance measurements that we might want to use for the compartmental modeling. That's done here. And also the bioavailability was asked. So we need to split up the PK parameters into the IV and PO route and rejoin them. So we have the 2 AUC measurements separated. And finally, let's just calculate the bioavailability by the ratio of the dose normalized AUC values for the PO and the IV route. So this is then goes together with the final parameters. So we stack up those parameters that we've derived. These are all for the fact sheet, and because it's 3 measurements, 3 different, we run some descriptive stats to get some mean values. And these mean values are then reported in form of a table. And this is already half of the analysis required for the fact sheet. So we are halfway done, but it's really quick to get. I mean, we just import, prepare, explore, and very quickly we are at the first result. And this was NCA and BA. We need to do some PK, some compartmental modeling, and that's because the tumor growth inhibition study data, they only have satellite PKs measured on the first and on the last day, so you can't derive any good estimates like Ka, volume, or clearance from those. So you need to do this from separate studies, and that's why we have this data in here. And here's the initial estimate step, which shows you the predicted curve and the observed data points in circles from the PO study. And what's important here is, the time unit has changed. It's no longer given in hours. It's given in days. And also the concentration is no longer given in nanograms per mil, but in micromoles per liter. And all of this has been taken care of in these preparation steps, so you don't need to worry. And you just need to adjust the initial estimates here in this display, run the fit, and take those final estimates and use it later on for the PK/PD modeling stage. So this was compartmental modeling. The final step is really PK/PD analysis. So we use a Phoenix model, and here we use a textural model. This is an adaptation of Simeone's tumor growth inhibition model, and unfortunately this is not a very simple model because we have to deal with 8 parameters. And that is sometimes a tricky problem. So first you need to find good initial estimates, then you have to fit those to your data and hopefully find a good final estimates from those. But here you see already, 3 parameters out of 8 we have already derived from our separate compartmental modeling activities from our PK model. So we just freeze those so they won't change when fitting the tumor volume data. But still, we are left with the estimation of 5 parameters, and that's also sometimes not so tricky, sometimes not so easy. What I did here is just start with an unperturbed curve, with the vehicle data that I've extracted, and just try to derive the first 3 PD parameters where you can get estimates for the linear and exponential growth phase. And here you can also adjust the settings here in this initial estimates tab, and when you're satisfied, you just run the fit and get some good estimates, hopefully, for the first 3 parameters that you then move over into this perturb model, where you then only have to estimate the remaining 2 parameters by fitting. And once you are done with this, you can then filter out the fixed parameters from this model. Also, we've got some secondary parameters defined, so these are also extracted from the model, appended into one single worksheet, and then prepared in a table format that you can easily report. For the fact sheet, actually, we have to filter out the K2 parameter, and as I said, K2 gives you a measure of the potency of the drug, so this is a very important parameter. But before getting there, you have to do some model validation. And here what I typically do is a sensitivity analysis, for example, for the perturb model, where we try to get good estimates for K1 for 2 parameters, K1, K2. You can apply some larger or smaller changes to the estimated parameter value and see how the likelihood changes when you apply those changes. And what you want to see is that you get to a minimum value of your log likelihood minus 2LL in your data, and that's what's shown here. And next step in validation would be a bootstrap, where you just resample your data from your observed from your study data and fit those, in this case 100 times, and try to get some estimates from those, and you just compare your final estimates if they are lying within those limits given by these bootstrap runs. So this gives you some idea about the robustness of your model and the validity of your final estimates. So this is all now the modeling stage. Once you're ready with modeling, you can start using those estimates to run some simulations. And simulations is you just apply some different dosing regimens, or where you apply different dose amounts. If you just apply different dose amounts, you just have to enter a value that you want to run against, and then in these two steps, you can create a dosing table by stacking up this against all the different animals, and you've got the dosing information that you can put into your model once again and run some visual predictive check like this one. And here it will give you the output with the blue lines giving you the limits of your predicted tumor volumes against the blue dots, which are actually the observed ones from your study data. Dosing amounts and dosing schemes. Here I make use of those different timetables that I've pre-prepared in my input data, and you just enter again the amounts that you want to apply within your simulation, stack up those doses for all your animals and days, and finally filter the scheme that you want to run. In this case, I want to run a QD study, 5 days on, 2 days off, and then I can apply this to the simulation. Again, a visual predictive check simulation where you see here the outcome. So this is simulation, and that's the main steps in my workflow. You see, you start with preparation, exploration, modeling, and simulation. All the steps covered in one single workflow. You've got lots of results coming with those objects in those sub-workflows, how to get the data out. And here we have a nice function, the reporter object, that allows you to extract those information. The first thing to get, for example, to the fact sheet is to append the PD parameters and the PK parameters that we've been given from different sub-workflows. Create a table object here for all the parameters, and this is actually the complete fact sheet for one compound. All the data given in the right format, in the right order. And in order to turn it into a Word document, there's this reporter function where you can pick objects from any of those workflows, and this shows you the object display, but what it also has, it has some context-sensitive information. So, for example, if you can apply headers and footers with some object-specific information, this is context-sensitive. So this object comes with information about some parameters, mean values, and the compound itself. And you can use those context-sensitive information to build up a caption that is specific for this object. Once you change some data upstream, like in the data or in the workflow sections, also the information in the final report will get updated, so reflects the proper state of the project. So this is more or less the fact sheet that is geared toward senior management. This is just very condensed, but when you want to get a project update for the project team, you may want to get more details around what you've done. So this reporter object also gives you the ability to prepare some layouts, some section headings and section breaks together with individual information like this. And all of this also with context-sensitive information. And the final level, the detailed report, this is more or less for your own files, so where you can really judge, how good your model was. So you can look at more individual data, not looking at sections of the parameters. You look at all the parameters And for the Phoenix models, you also want to see lots of more diagnostic plots like these to make really sure that, yes, this model was good, and so you can do with that. So this is showing you the reporter function. That is the compound-specific workflow. But what I said in the beginning, I mean, you can also do a workflow where you can compare across compounds, like what we've seen here. One way to do that is just to import all the, individual compound workflows into a single project, like I did here. So you've got now 3 different workflows, and they also contain the same sub-workflows, the substructure, and the same information, but now for 3 compounds. And if you look at the report, you can now extract all the parameters from the individual compounds and get all for the individual information from each compound and create a parameter table here where you can see the fact sheet for all 3 compounds. And that is exactly what we have been looking for when I started the presentation. It's the same as with the summary report or the detailed report. It's much the same. You can also collect all the information from all the individual objects. An interesting function that I wanted to show you is also the model comparer object. The model comparer object is an easy tool to organize analysis or results from all the different models that are reside in your workflow. So here, what you see here is a list of all models, Phoenix models that have been generated in this project, and you can pick those that you really want to compare with. So I've just picked the perturbed models like here and here for the different compounds. And the next thing you can pick is for the sheets, the worksheets, which columns you want to see in those worksheets and which worksheets and which plots. And if you look at the results, here's just the comparison result, the overall result with the objective function value compared across these compounds, theta values, omega, and so forth. But this is a very nice thing. You can get also different plots. So when you, for example, want to see the fits of the data, you can look at this, and you see this is for the one compound, this is for the vehicle, EL102, the second, and the third compound, where you can simply look at this across the different compounds. Okay, that's all what I wanted to show with my software demo. Let's go back to the slides. Okay, so here's once again the overview of the workflows that I presented. And I showed you how you can easily prepare your study data to be ready for modeling, because modeling is really the crucial step. And at the end, you want to get the best models with the best parameters out of this workflow. And what I've shown you, or what I want to focus on the next is there are 2 operations that are very important to save time in this process. It is the data in and the data out operations. So if you look at the data in operations, you've seen importing the study data from, for example, in this case, Excel was straightforward. You just import it right away. And then there are just a few steps in preparation, but these are really highly efficient because they really get you the data into the proper format ready for Phoenix analysis. And this will always be required for any data when you want to automate things. And for this workflow that I've shown you, the whole workflow was automated more or less. And in this particular case also, we have been able to generate the tumor volume data together with the dosing tables. And can you imagine how cumbersome it would be, and also how error-prone it would be if this would be done manually? I mean, that's needs to be taken into account. So, for the data out operations, I mean, we have been looking at the reporter function, and this is just a very efficient way of dealing with all the plots, tables and listings that are spread across the project file in all these different workflows and sub-workflows. So you can easily extract and collect them, and also to provide the proper layout and create a single report document. And this can be geared towards specific audiences, as I showed, for either senior management type of reports or project update reports, whatever is required. So the function is very flexible. But whenever you change something upstream, like the input data, or you change some objects, the reporter object will get invalidated, will get pink in color, like any other Phoenix object once the underlying data is changed. And all the dependent objects will also get invalidated. But just by clicking on the refresh button of the reporter object, it will know about all the dependent objects upstream and will update those objects first and finally give you the ultimate up-to-date data report. So what I next showed you was the model comparer function, which is also a very important function. Is extremely helpful whenever you need to compare several models, or in this case, several compounds. And it will simplify the comparison by organizing the diagnostic plots, or also tables, of the models next to each other. So you can easily see what is going on when you switch from one compound to another, or when you change from one model to another. So let's see, and now we're coming back to our fact sheet from the beginning. I hope you now have a better understanding how you can quickly and efficiently get estimated preclinical parameters into your fact sheet. In fact, if you're using the standardized workflows that I presented, once you update your data, you will be only one click away from your final report. Everything is being handled automatically. So with this, I'm at the end of my presentation. But before that, I just would like to say thanks to people that helped me on this way. So there's the Elara team, of course, and the team at Certara. And with that, I want to thank you for your attention and hand over to Suzanne for question and answers. Thanks.
Okay. Thank you, Bernd, for that very helpful presentation. It looks like we've got a good number of questions from our audience. First person wanted to clarify that the preclinical data was from mice. That's correct?
This was, yes, xenographs. These were in mice. These were tumor volume measurements in mice.
Got it. How would you estimate human parameters from the mouse xenograph data?
I mean, there are some allometric scaling that you can do in any case. I mean, it's maybe a little bit beyond the scope of this session. I mean, when we are working on Phoenix workflows to go through the individual steps of allometric scaling, but this is just another workflow, if you will, that you can add to the to this workflow. If there are specific interests, I could get back to this at handy, maybe by personal email, because it's a model-specific question, I would say.
Also, I would add to what Bernd said and let our audience know that we'll be having a webinar on allometric scaling with Dr. Serge Guzzi and Tin Nguyen in March 17th. So if you are particularly interested in learning about allometric scaling, I invite our audience to register for that webinar. Another person in our audience wanted to know why the doses were entered in micromoles per kilogram, and he wanted to know whether we can use milligram for doses and keep the concentrations in milligrams per liter.
Yes, you can. I mean, whatever unit you prefer, you can stick with those. That's right. But sometimes, I mean, especially when you compare these results with in vitro data, it sometimes makes more sense, I mean, to have this in micromoles. This is really up to you. I mean, I've shown you that this is part of the preparation where you change from nanograms per mil the concentration as well as the dose into micromoles. But this is up to, this is something that's an optional conversion, if you will.
Customer from our audience would like to know whether the reporter object from Phoenix Connect that you demonstrated, whether that can present results in a lattice format. You
can arrange the plots in the reporter, I mean, in a lattice format, if you want to do that. There are several ways how to deal with it. Either you have already prepared a lattice in Phoenix and you take over these, or you have created single plot object page plots, if you will, in Phoenix, and arrange this through a lattice format within reporter. There are two options available here.
Someone would like to know how do you change the workflow when the format of the study data changes? For example, when you have data from a different CRO.
Oh yes, this is really simple because you have, or I have to say, several templates from each of the sub workflows. So, for example, for the preparation, I've got specific preparational steps for each CRO that we've dealt with. And these are more or less small changes there because of the different Excel formats that they use. But it's more convenient and less error prone if I just use the template that I already worked with. So I import the CRO-specific preparation workflow. And the same, applies also to the model. I mean, sometimes we don't use the Simeone model. Maybe we use a Gompertz model or so. I've got a specific modeling workflow that deals also with some other models that you can easily import as a template and, just link the data to those workflows, and then you can run it very quickly, efficiently.
Someone else would like to know whether whether you need to have a license for Phoenix NLME to use these models, or whether some estimates can be calculated with naive pooled population analysis.
Yes, I mean, yeah. What I've shown here was population mode. So for all the models were run in population mode, and that means you would need an NLME license for this. But you can also do it in a two-stage approach, where you just run naive pooled engine and just do some descriptive stats on the results, and then you have similar kind of results. I mean, it's just a different approach. I mean, people would prefer the population mode, but if you want to run naive pooled, that's up to you. It's open. You can do that using the two-stage approach.
Similar question along those lines. Someone wanted to know why you chose to use a population modeling approach.
This is a preferred approach, I would say, because you've got lots of data, and you really want to get. I mean, there's some variability, and you just want to really get a good value out of it. From the whole observation. There's a large number of observations. You will get a good result out of that, and the preferred approach is actually the population mode. Because it looks where variability exists. It could apply this to either the random effects or residual variability, and I guess this is a proper way to deal with data of this kind.
So we talked about how you used both Phoenix WinNonlin and Phoenix NLME for the steps in the workflow. Are there any other licenses you would need to perform all the steps that you showed in your workflow?
Yes, as the other attorney mentioned, I mean, for the reporter function, you would need the Phoenix Connect license. But also this, I mean, of course, it's optional. I mean, it's a nice to have, and it's really a saving time tool and a time saver tool, because it allows you to create documents, like you want to readily submit to your colleagues or to your boss or whatever. So it really is. If you want to save time, you need to use the reporter function. If you want to prepare your Word documents on your own, you create those objects in your workflow, and you just have to export them into a Word document and deal with the format separately.
This looks to be the last question from our audience. Is it possible to create the reports in PowerPoint?
Actually, the reporter function does not allow to export into PowerPoint, but I guess this is part of our product backlog list. So we have envisioned to support that in a future release, but we actually lack some feedback from the customers. So in order to really make sure this is a tool or this is a function that many customers will use. So if you think this is important, please let us know, and this will feed back into the product backlog prioritization, and perhaps that in the next release we will be able to release that.
Looks like we have one more question coming in. Can the model code you used be derived using the graphic user tool available in Phoenix?
No. I mean, it would be too yes, it would be too cumbersome at this stage to deal with all the different formulations. The graphical user interface gives you a lot of flexibility. You know, you can create parameters, you can create expressions and procedures, and this will be a little bit too disconnected in the graphical user interface. So I guess in this case I would go with the textual right away, because it's much more easier. The PML language is not too difficult to understand if you have modeling experience with other programs like SAS or S or R or whatever. So it's straightforward to use, and you should be able to handle this. It's really just a few lines of code, as you've seen. It's not very many, and so it's more efficiently to be done in textual mode rather than graphical.